Assalamualaikum dear students. I welcome you to the lecture number 43. Here I am, Miss Sana Ismail, lecturer of botany and working as a co-teacher in the Department of Botany at the University of Education, Lahore. Today we will going to discuss one of the topic from the subject Plant Systematic Anatomy and Development. The course code of this subject is BOTN 1112. The topic we will going to discuss from your course content is Parthenocarpy. Let's start our lecture. Parthenocarpy. Parthenocarpy is the growth of ovary into seedless fruit in the absence of pollination and fertilization. It may occur naturally or can be induced artificially by exogenous application of hormones or their enhanced endogenous level. Parthenocarpy improve the yield and quality and processing attributes of vegetable crops like cucumber, eggplant, and watermelon, where seed is a limiting factor during consumption. In this slide, you can see here the two pictures. On the right side, there is a Parthenocarpic example in which no pollination and no fertilization occur. On the result of this, their fruit development will be seedless. You can see here the structure of a fruit where seedlessness of fruit are present. On the other hand, when the pollination and fertilization occur, then the fruit and seed development are properly performed. Parthenogenesis. Parthenogenesis is a process which refers to the production of egg cells without fertilization. For example, mangosteen. This Parthenocarpy trait provide highly useful to develop fruit under environmental conditions that are unfavorable for successful pollination and fertilization, particularly in greenhouse cultivation and especially in the cross-pollinated crops. It is an established fact that phytohormones play important role in fruit setting and their genetic manipulation can lead to seedlessness. Therefore, this present lecture is focused on the factor. Here is an example of Parthenocarpic fruit and which is comparison with fertilized fruit. In section A, the fruit of wild and seeded plana, Monza acuminata, which contain fertilized seeds, developed as fertilized seeds. On the other hand, in section B, the domesticated Parthenocarpic banana fruit, which are seedless fruit. In the section C, here is a fertilized induced tomato fruit of the Monalba variety, which are fully fertilized and pollinated and which induced a seeded plant development. In the D section, when this Monalba wild type fruit cut in half, the locule is filled with pulp and the seeds are clearly visible in this section. On the other hand, in portion B, a parthenocarpic fruit from Nelbo containing the abnormal ARF8 mutant transgene. When we cut into half portion, then we can clearly see that in their central columella, which is represented with C, is enlarged as compared to the Monalbo, which are fully fertilized. And here the locule is filled with pulp, but there is no seeds are present and showing a seedless endocarp. This is the comparison between parthenocarpic fruit. So here is another example of seeded fruit and seedless fruits. In this picture, the section A represented Ananas squamous. Their common name is custard apple, sugar apple, or sweet soap. In Pakistan, it is widely cultivated in Sindh and also in the Punjab. The fruit is the best tasting of all the ananas species. Their pulp is said to be rich in vitamin C. You can see here in this picture, on the left side, the seeded fruit are present, while there on the right side, the non-seeded or seedless fruit variety are also present. It means it is an example of parthenocarpic fruit, which are seedless fruit. In the section B, the example of Actinidia arguta, their common name is hardicavi. On their upper side, the seeded fruit are present, while on the lower side, the seedless fruits of variety are present. In the section B, the Cucumis sativas are also exempled. Their common name is cucumber. On their left side, the seeded fruit variety are present, but when the proper pollination or fertilization does not occur, then their parthenocarpy process are started and the seedless fruits are developed. In the section D, the Elias oleifera, their common name is African oil palm, in which if the seeds are fully fertilized and pollinated, then the seeded fruits are developed, while when the fertilization does not occur properly, then the seedless fruit are setting. In the portion E, the citrus clementine, which are also called the Mandarin orange, you can see clearly in this portion the seeded fruits are present. Seeds are present in it. While when the proper fertilization does not occur, then it may be naturally processed or may be artificially processed, in which the seedless fruit are developed. The seedless fruit are desirable for improving the quality of fresh as well as of the processed fruit, and it has been observed in cucumber, eggplant, watermelon, tomato, and in the orange plant, in the oil palm, and so many vegetables and fruit varieties. Hence, it may be depend on the genetically tool of Parthenocarpy, which play an important role in it. In which Parthenocarpic, the ovary developed into a fruit without fertilization, and the fruit growth from fertilization and seeded development result into a seedless Parthenocarpic fruit. And this Parthenocarpic is genetically controlled and commercially exploited for the production of seedless fruit. And Parthenocarpy are divided into two categories. The first is artificially or induced Parthenocarpy, which involve in the stimulation for the growth of a fruit using both natural or artificially plant hormones. The induction of Parthenocarpy is a common agriculture practice for some horticulture species, while on the other hand, the second category is the natural or genetic Parthenocarpy. The natural or genetic Parthenocarpy is also called the obligatory when expression of the Parthenocarpic trait is not influenced by external factor and is facultative. It is also called the facultative Parthenocarpy if it occurs only under adverse conditions for pollination and fertilization. The genetic Parthenocarpic in some plants act as a vegetative Parthenocarpy. During this Parthenocarpy, plants that do not require pollination or other stimulation to produce Parthenocarpic fruit, then they have the vegetative Parthenocarpy. The genetic or natural Parthenocarpy is also called as stimulative Parthenocarpy in some plants, because during this procedure the pollination or other stimulation is required for Parthenocarpy. That's why they are termed as stimulative Parthenocarpy. Stenospermocarpy. The Stenospermocarpy may also produce apparently seedless fruit, but the seeds are actually aborted while they are still small Parthenocarpy. It usually occurs as a mutation during reproduction process. The Stenospermocarpy, they are immature seeds, and the watermelon, seedless watermelon, is the example of Stenospermocarpic Parthenocarpy. While the cucumber are an example of vegetative Parthenocarpy, and the banana. So here in this slide, the triploid banana fruit, which is an example of vegetative Parthenocarpic process. There is a graph of growth in volume of seeded banana fruit are sigmoid in shape. Those of Parthenocarpic fruit are variable but are not sigmoid, and the shape are related to specific origins. Growth rates are related to certain ovule behavior, to seed content of the fruit, and to ploidy. Here is an example in which the triploid banana or hybrid banana are formed by crossbreeding of Mosa acuminata and Mosa albiciana. By crossing these two Asian species or varieties, the hybrid banana are formed, which are Mosa paradisea. The origin of triploid banana are formed from these two hybrid varieties, in which the one variety give one haploid set of chromosomes from Mosa acuminata, while the hybrid banana get the one haploid set of chromosomes from Mosa albiciana. As a result of this, the triploid banana variety occur, which are sterile in nature. It means they doesn't contain seeds. That's why they are called the seedless fruit, and their process are called parthenocarpy. During this, the naphthalene induroacetic acid induces parthenocarpy in seeded banana and stimulate it in weakly parthenocarpic types. By contrast, coumarin, a hormone inhibitor. So, when successful pollination and fertilization does not occur, then development of seed cannot perform properly. That's why the fruit becomes seedless, which are also called the parthenocarpic fruit or seedless fruits. If the pollination and fertilization occur properly, then seed development performs successfully. As a result, the seeded fruit or pollinated fruit are produced, which contained a number of seeds. So here in this slide, the example of Stenospermocarpic grapes, in which the normal pollination and fertilization occur, but the embryos abort when they are young. Often the remnants of seeds can be seen in the fruits. The characteristic feature of the group of Sultana type raisin cultivars, for example, Kishmish, Rosobi, which their common name is pink sultana, black kishmish, black manuka, and Thompson's seedless grapes are the example of Stenospermocarpic fruits. Origin of Parthenocarpy. The Parthenocarpy originate due to absence of pollination and fertilization. It is also originate due to male sterility, due to degradation of pollen mother cells. The Parthenocarpy also originate when the fertilization of ovule degrade, and the Parthenocarpy also originate due to self-incompatibility, for example in the citrus fruits, and the Parthenocarpy also originate due to embryo abortion, for example in the grape, which the process are called stenocarpic parthenogenesis. The parthenogenesis also originate due to chromosomal irregularities during meiosis, which leading to triploidy, and the example of these are the triploid banana, which are occur due to vegetative parthenocarpy. And other examples are Oro Blanco and Malacold are the example of parthenocarpic fruit. In this slide, the physiological basis of parthenocarpy would be discussed. To obtain parthenocarpic fruit is a physiological challenge. Fruit development comprises early development and maturation. Early fruit development in most of the plant involves three phases. The first phase is fruit setting. The second is cell division. And the third and last is cell expansion. The ovary takes decision in the first phase. Ovary takes decision to either abort or to go further in fruit development during the first phase. If the pollination occurs properly, then the fertilization starts and the fruit are further proceed into seeded plant, seeded fruits. And if the pollination does not occur properly, then the fertilization are also cannot properly performed. That's why the fruits become parthenocarpic. It means it depends on the ovary to decide either abort or to go further in fruit development. During cell division phase, which is the second phase of fruit development, during this phase the increase in fruit size is slow because the dividing cells are small and tightly compressed, and the number of cells decide final fruit size. If the fruit size are large, then the fruit became fully developed. But if the fruit size or seed form are low, then the seed formation cannot properly occur. The third and the last phase begin after cell division stops. The fruit grows by the increase in cell volume until it reaches its final size. The cell expansion commonly increases fruit size by 100-folds, and this makes the greatest contribution to the final fruit size, and the embryo maturation occur, which are further developed and the fruit are ripened. If these three phases occur properly, then the fruit are fully matured and fully developed. On the other hand, if the ovary are abort and the embryo development does not occur properly, then the seedless fruits are formed, which are called Parthenocarpy fruit or Stenospermocary. So here is a comparison between fertilization, Parthenocarpy, Stenospermocarpy and Apomixis. During the fertilization process, the proper floral development or fusion of gametes occur. It means the successful pollination and fertilization are performed. As a result, the embryo maturation, formation of endosperm and integument growth proceed in a successful way. And at the end, the fully developed fruits are obtained. While in case of Parthenocarpy, the floral development occur in a proper way, but the ovule development cannot perform in a proper way. That's why the post-pollination occur, and as a result, the seeds are obtained, fruits are obtained without seed. In case of Stenospermocarpy, when the floral development occur, then the pollination and fertilization are also performed in a proper way. But after fertilization, the defects in embryo appear and the ovary may be abort. That's why the endospermic development cannot occur in a proper way, and as a result, the fruits are obtained without seed. And in the case of Apomixis, during Parthenocarpy, is the formation of fruit without fertilization, but the apomixis is the formation of seed without fertilization. It means during apomixis, the formation of seeds cannot performed in a proper way. Means when the seeds are produced, the fertilization are not occur. That's why the autonomous fruit and seed development are performed, and the autonomous fruits are obtained during apomixis. This is the comparison between. So in this slide we will going to discuss about the factors affecting or influencing parthenocarpy. Environmental factors. The adverse environmental condition inhibit fruit set and growth of several vegetable crops. High or low temperature, humidity, low light intensity, heavy rains, and strong winds are all factors which negatively influence several steps of the reproductive process, such as formation, dispersal, and germination of pollen, fertilization, and seed maturation. The fruit production due to these factors, these environmental factors, is impaired. Parthenocarpy is also affected due to chromosomal aberration and due to genetic disorders. There is another factor that influence parthenocarpy. These are the growth regulators. These plant growth regulators except ethylene and abscisic acids induce the fruit development. However, the gibberellins, auxins, and cytokinins induce parthenocarpy. The self-incompatibility also influencing the parthenocarpy, while the genetic factors like gene controlling meiosis are also influencing the parthenocarpy. These are the factors that affect the parthenocarpy and create the parthenocarpic fruit and vegetable crops. In this slide, we will going to discuss about the role of growth regulators in parthenocarpy. The growth regulators are auxins, gibberellins, and cytokinins, or the mixture of these are effective in inducing fruit in several crop species. Auxins and gibberellin may act in parallel or in a sequential way on fruit set. Auxins may be natural or artificial supplied exogenously to unpollinated flower, which induced fruit growth in tomato and in other horticulture plants. It can replace the signals which are provided by pollination and fertilization. Gibberellins activate, which are able to induce fruit set parthenocarpy. It increased the level of gibberellin in pollinated ovary and an increased expression of gibberellin's biosynthetic genes. Next we talk about the ethylene. In unpollinated tomato ovaries, ethylene biosynthesis and signaling genes are highly expressed, and it is known that in many plant species, pollination itself triggers a transient increase in ethylene production. It plays a role in coordinating ovary growth and flower senescence events that are associated to fruit sets. On the other hand, if we talk about the brasinosteroids, it interacts with auxins, and the partheno growth in cucumber, if we discuss about it, then it activates their cell division, and if we apply the brassinosteroid inhibitors, then it blocks the fruit growth in a parthenocarpic cultivar. Importance of parthenocarpy in vegetable crops. Parthenocarpy is an important trait for improving the yield, quality, and processing traits of vegetable crops. This trait proved highly useful in greenhouse cultivation, particularly cross-pollinated vegetable crops. This is established fact that phytochrones play an important role in fruit setting. However, their genetic manipulation can lead in the development of seedlessness. Further exploitation of biotechnological tools can enhance the efficiency and identification of parthenocarpic genes across the crops for the benefit of mankind, such as we can get the off-season production of fruit and also get the low cost of cultivation. So this is all about for this lecture, and here is the reference that are used during the preparation of this lecture. Thank you.